and gentlemen, for those who are online and also in the room, welcome to join this State Key Laboratory of Marine Environmental Health Distinguished Lecture. So we are really honored to have Professor John Gissi with us. So I'm going to introduce him to you. I need to read my script. Before I give the details, I would like to highlight that our state key lab was formed officially in 2010, and then before that we had a team called Area of Excellence Center for Marine Environmental Research and Innovative Technology. During that time, John already with us. He was appointed by City University of Hong Kong as the professor at large, very, very big professor for many years. Then he educated many PhD students and master students over time, and also helped some young professor like me. At that time, I was still a junior faculty, but John gave me lots of advice, and also since then we worked together on various projects. Every time I learn a lot from him. He is not just knowledgeable, but he is really, really passionate about environmental health, and also he is really good in the field in environmental chemistry, to the how to frame the research questions, how to think about the new technologies, and then he also worked with many different peoples in the world. Then he knows the subject development really well. He can tell you where you should go for, what is the next wave. Then later on, you will have a chance to discuss with him. He may give you some tips for the lottery tonight. So, Professor John Gissi is an American-Canadian ecotoxicologist, very famous for his groundbreaking work in environmental toxicology and discovery of various harmful substances in the world, and also giving advice on how to minimize their impact. He is currently a distinguished professor emeritus and former Canadian Research Chair in Environmental Toxicology at the University of Saskatchewan and Michigan State University. He was the first scientist to discover toxic PFAS in the environment, means perfluorinated compounds. His extensive research also includes photo-enhanced toxicity, endocrine disrupting chemicals compounds and their impact, and more recently, the fate and transmission of SARS-CoV-2 virus, particularly regarding its persistence in wastewater and the environment. With over 1,300 peer-reviewed publications, this is amazing. With an H index of 151, he is among the world's most cited scientists in ecology and environmental sciences. He has been elected as a Fellow of Royal Society of Canada in 2010. He has received many, many awards, including the Einstein Professorship Award by the Chinese Academy of Sciences. Without further ado, please join me to welcome John. Thank you. You can sit and talk if you like.
I'll stand for a bit.
Okay.
See what happens.
You can either use this mic or say it. Oh, that doesn't work. Okay.
It's not a very big room. Maybe you can hear me. Then I.
One I wanted to listen.
Oh, okay. I'll hold this then. Okay. So I'm a chemist. When I was young, when I was your age, I couldn't decide if I liked chemistry or biology better. So I've always sort of done both. Where's Henry? Henry. So, well, and Rudolph is here, a longtime good friend. But Henry was a student with me and worked on some very complex problems. So we always get the hard problems, and we always try to use chemistry and biology together. But today, I'm going to talk about something a little different. I'm going to talk about wastewater-based surveillance of COVID-19. Oops. So I have a lot of people to thank, and I have to give some background on how we started to do this. So first of all, we were doing studies with wastewater treatment plants. So down here at the bottom, Mike Sadowski is at the wastewater treatment plant in Saskatoon, and we had projects with them. There were new laws coming into effect in Canada about wastewater quality and what they needed to measure beyond the traditional things that they had been measuring. So he had met me and asked me if we would help him. So we were doing that. And in the process of doing that, we had the idea that we would look for things like recreational drugs. So we had a project, and now Henry's working on that as well. We had a project with the Royal Canadian Mounties. It's our state police in Canada. And so we were helping them. And I had applied for a grant because we were interested in eDNA. So one of my former students, Zhang Xiaowei, is also a world expert in eDNA. And when he was with us, we were developing this to do things like monitor for invasive species, to monitor for endangered species. And so we applied for, and I have to go back. So down at the bottom here, you can see we had a program called Environmental DNA Global Water Futures. And I applied for a big federal grant in Canada with my really good friend, Mark Servos. Mark Servos was at the University of Waterloo, and I'm at the University of Saskatchewan. So we wrote this big proposal, was for 87 million Canadian dollars. And Mark and I were the co-principal investigators, and we organized all of this. Now, we had agreements with the government. We were developing techniques, for instance, to monitor all over Canada for zebra mussels, a gift from you in China to Canada, which is a real big environmental problem. We were monitoring endangered species like tree frogs. With global climate change, all the surface water ponds where I live are drying up. So it's a real problem. So anyway, we had this. We had agreements with many different provincial governments to help with various monitoring programs. We had agreements with industries to develop eDNA and compare to their traditional. So we had a lot of things planned. And then what happened? COVID happened. Everything stopped. Okay, the university was closed. We weren't allowed to go 2 people in a car for sampling. You couldn't have more than 1 person in the lab. And so all of these problems really killed. And our cooperators, the industry said, oh, don't come here. The government said, oh, we're closed. You can't do anything. So what to do? I had all these students, postdocs we had recruited in the area of eDNA, and I had $87 million. What to do? So Mark and I thought about it a little bit, and we decided that we would try to develop something to monitor for COVID. I got COVID for the first time in March of 2020. So it was a personal kind of a thing. Anyway, this is sort of my team. You may know some of these people, but this was the group that really developed the work. This was our first paper. I don't know how many times it's been cited. Kenny probably could look it up. But this was our first paper published, and it was a cover article in ES&T Water, and got a lot of press around the world on how to do this wastewater monitoring. So I do a lot with chemicals, and what I'll tell you is chemicals will make you sick, but bacteria and viruses will kill you. So it's a big concern, but it was a a change for me. It was not the usual kind of thing that I would be doing. And so I put viral diseases right up there as a really important kind of environmental effects. Now, in addition to that, there's a social economic side to this as well that I'll come back to at the end, but I need to tell you about now. In Canada, we have indigenous populations. And in fact, in my province, we have 17 sovereign nations. Okay, so these are countries within a country. And these are native Canadians. We refer to them as indigenous persons. And they were there long before people like me, Europeans, colonized. But when that happened, when Europeans colonized Canada, they brought with them diseases. And in particular, the disease that was the biggest problem was tuberculosis was one, but smallpox. Smallpox decimated these populations. And what's worse, where there were unscrupulous people that were actually giving contaminated blankets. So the native people liked to trade furs and things they could collect for manufactured goods like steel and pots and pans and guns and blankets. And these people gave them blankets that were intentionally contaminated with smallpox. Terrible, just absolutely terrible thing. So in the end, we'll come back and talk about those populations and how I was able to help them and their distrust of the government. Okay, because of this historical event, they were very, very distrustful, particularly when the government told them they were there to help them with an infectious disease. So there's a whole book about this, Guns, Germs, and Steel, which I highly recommend. So another reason to talk about this is I have a personal connection. So in 2002, what happened in Hong Kong? Rudolph? SARS, right? I was here. I was living here. It's Rudolph's fault. He invited me and recruited me to be here, and I came, and I was here for SARS. So that was an interesting experience. I learned a lot. And the other thing is, if you look here, this can you see this? Move this around? No, yes, no, no, maybe not. So down here, the Spanish flu. So I have a personal connection to that because both of my paternal grandparents, my father's parents, died in that epidemic. So between living here in SARS and having my ancestors die because of this, I have a personal connection. Timeline, but things basically started in March of 2020. And on the bottom here, I have what we did in our laboratory to develop these things. And so we had a number of benchmarks as we moved through. But we had to do things very, very quickly to try to have an impact on what was happening. So a little bit of background on COVID-19, if you don't know. The big key thing about it is it's an mRNA virus. It's not a DNA virus, so it's mRNA. We were doing eDNA, so we had to switch things to do eRNA. RNA is a lot harder to work with in the environment than DNA. It degrades more. And as I work through this, I'll tell you what we had to figure out and how we had to do that. There are a number of these coronaviruses that infect a wide range of animals, everything from bats to mice to beluga whales. The big white whales we have in Canada can be infected by coronaviruses. So it's a wide group of viruses. Oops, sorry, I keep going the wrong way. A lot about the virus because it was such a big epidemic and so many people around the world were working on it, we really knew quite a bit. On the left-hand side here, there's an NSP1 and NSP2. Those are structural proteins in the virus, and that's what I used to monitor the virus. The spike protein is important and was evolving and mutating and changing, and that was leading to different strains of the virus. And so I also monitored the spike protein to be able to determine which of the strains of the virus, whether it was Omicron or Delta, the different ones that developed. Okay, so those are the key proteins. Now, I was on 4 different panels, global panels, and I was the lead person for Canada. And I worked closely with our National Health Service and Health Canada, and also with our National Microbiology Lab in Winnipeg. But I was the chair of the committee, and we had subcommittees for methods development, and everything even through ethics, the ethics of doing this kind of monitoring. And we've written reports on all of this, and they're available. If you're interested, I can provide them to you. Okay. Sweet. A bit of background on the virology. So it's a respiratory virus. So you're infected really by breathing in particles, but then it can move throughout the body and cause a number of different effects, including lethality. So while it's developing, it resides in the nasal passages and then throat and then lungs. And ultimately, one of the key things is that it then can be excreted in waste. So you can find it in feces, and that's what we're going to be monitoring. A little bit of background, things came on very, very quickly. A lot of people around the world got sick, so there was a lot of pressure for us to come up with answers. So in Canada, one of the problems, if you don't know about Canada, we're the 2nd largest country in the world. Okay, only Russia's bigger, China's smaller. I think China's 3rd or 4th, maybe Brazil's in there somewhere. But we're the 2nd largest country in the world. So land area wise is very, very big. And we're only about 39 million people. More people live in Texas, more people live in Beijing than live in Canada. So we're a big country, long distances among cities. My daughter lives in the Calgary area. It's a 9.5-hour drive, and that's the next major city. So it's a big country with not many people. And that means we don't have the tax base for a lot of things that you can have here, like public transportation. And our medical system we have universal medical care, so it's free for everyone, but it's really been under pressure and limited, and we have a shortage of doctors, shortage of nurses, shortages of hospitals. So when SARS hit and when COVID hit, it was a really big problem. And one of the issues was we did not have enough beds for the people that were sick. So the government didn't have enough respirators. And so they were moving people around this big country to try to get the medical help that they needed. And remember, we have all these sovereign nations, these small communities of indigenous people that live in very remote areas. And many of them we can't drive to. We have to fly to get there. So big country, small population, a lot of people getting sick, a lot of concern, a lot of fear, a lot of confusion, and a lot of distrust of the government. And they put all this on my shoulders to try to figure out. So the first thing is, how do we diagnose COVID, and what is the etiology of this disease? So the first thing is you get some physical symptoms, and those can be by a self-check or by a physician, but you can be infected long before you express any symptoms. And in fact, some people get very mild cases, and some people have no symptoms at all. So they're carrying the virus, they're transmitting the virus, but they're not really sick at all. So they don't show up and say, I'm sick. They don't show up to be monitored. So this is moving around through the population, and people are getting sick. There are a couple ways you can monitor for this. You can do blood samples. You can measure antibodies in the blood. That's pretty invasive. And really, those antibodies don't show up until quite late in the infection. To measure is with nasal swabs. So we take a swab of your nose, and this is the traditional way that doctors would do the monitoring. So we would take that, and its advantages, it's directly related to individuals. But remember now, there are people that don't feel sick, so they're not reporting yet. So one of the things the government was doing was opening monitoring systems where you could drive your car. It was like drive up fast food. You could drive up and they would take a swab. There's a picture here with the guy getting a swab done. But then once they have that, it's several days before they can process the sample, because they would be doing what we would be doing in the laboratory, using extensive quantitative PCR to do this analysis. So it takes some time. So there's some disadvantages to this. Some people are asymptomatic. Some people don't report the illness. People that are worried about being told they can't come to work, or that their kids can't go to school, they don't report if they're sick. They go anyway. There was an availability limit of tests. The government was making as many as they could, as fast as they could, but they were limited. They ran out. And then we had a problem with one company that made a lot that were defective. They didn't work at all. So now we were getting false negatives. People thought they were okay, but they weren't. So there was the problem with the limitation. There is a delay in testing, a couple of days, like I told you, and then another delay in reporting results. It's semi-invasive. Has anybody had one of these COVID tests? Yeah, it's not fun, right? When they stick this thing up your nose and wiggle it all around, it's not really painful, but it's uncomfortable. And so it's semi-invasive, and it takes time and effort of individuals to go and do it. And the cost was huge. The personnel just to be there to do this testing. So there's some limitations. Oop, why do I keep going the wrong way? Okay, so one of the key things at this point is, where can you find the virus? And one place you can find it is in feces, human waste, stool samples. So we can get that. On the right here, I'm giving you some information about the timeline. So for instance, there'll be a peak viral load about a day before you have symptoms. So that means you're going to be sick and transmitting the virus at least a day before you know you're not feeling well. And then I've got information here on when the virus is being shed. So for instance, the upper respiratory tract, it's for 17 days. So it's a long time. So some of the guidance was, if you get sick, you basically have to stay away from other people for at least 2 weeks. Okay, and then we've got stool samples, and people will be shedding the virus into their feces for up to 17 days. And so I've got good information that we've gathered on this, on when it occurs, when the peaks are, and that'll become important later. How does that come into play? One of the things that I had against me was doctors and nurses did not believe in this technique. They were heavily invested in the old way of doing it. The nasal swab, right? Then you do the test, and then you get the data, and then they would put it into their epidemiological models. And that was taking anywhere from 10 days to 2 weeks. Okay, keep that number in mind, 10 days to 2 weeks. Okay, so that means once they got the data, they were 2 weeks down the road before they could affect any kind of control and decide what to do. And it was also a problem for the hospitals for allocating resources. Because now, by the time the peak hits, it's kind of too late to move respirators around the country. Big country, remember? Really big country. So we were doing wastewater monitoring for other things, for contaminants, for regulations, for the quality of wastewater. We were doing work with some of the work you do here with antioxidants in automobile tires. We were doing that in wastewater treatment plants. We were looking for pharmaceuticals. You can monitor the wastewater for the general health of a population. I can measure acetaminophen as sort of an indicator of general health in a population. I told you about recreational drugs, alcohol, tobacco. These are all things the government's interested in because it's hard to get data. You know, they can do a survey or ask people and they're not going to say, yeah, sure, I'm doing cocaine. They don't tell you that. So this was a good way. And in fact, we worked up methods that I can get you within a block of a coke house. So where they're making illicit drugs by making measurements in lift stations or from the wastewater sewer collection distribution, I can focus right in on where they're coming from. So we were doing this and helping the police to do that. So that's sort of what we were doing, and now we could apply this to wastewater-based epidemiology. So now we're going to switch and look at the viruses. So what can you do with this? Is we're going to monitor an entire community. Now, I implemented this at our university, and many, as you'll see, many universities around the world, maybe here at CityU, I don't know, implemented this kind of monitoring. So we're going to monitor a whole population, and then sample of that entire population in the wastewater. So everybody is excreting to the wastewater system, and so we're integrating the entire population. Now, we can get samples within the wastewater collection system at different places and focus in on different things. We can focus in on neighborhoods, and we can focus in on schools. We can focus in on hospitals. We can focus in on old age care, long-term care homes where we were having real problems with people. And then we can extrapolate this to a whole population. So some of the ways we did this at the university was we monitored the effluent coming out of a dormitory. Why do we do that? We've got 350 students living in the dormitory. So one way to monitor them every day would be to go in and do a nasal swab on every student. What's wrong with that? Any ideas? My family? Vivian, what do you think? Doing that for all 250 students every day. Personnel, and it's disruptive. And the students basically all have to be locked away until you can get the test results, and it takes a couple days. So it's really disruptive. It's a problem. What we could do is monitor the whole dorm. As long as it was negative, we let everybody go out and go about their business and not have to do a nasal swab and not have all that expense and time. If we got a hit for that particular dorm, then we would go in and test all the people until we found the people that were infected and isolate them and treat them and give them the medical care they needed. So it's very, very efficient. I was talking to my friend Brian Brooks, who's a good friend of yours here, and I'm also a professor at Baylor. So in the wintertime, January, February, March, I'm a professor in Baylor, and that's when the viruses are the biggest problem. And so I was talking to Brian about this, and he says, wow, we should do that here. And so we did. We started the program at Baylor. And from these committees that I was on, in the end, I think around North America, 250 universities started this kind of monitoring. You can monitor airports. So if you're concerned about people moving around the world and being infected, the alternative would be to test everyone when you get on the plane, and that's really hard to do. So it's not as effective to do airports, but it's something we talked about with the government that they possibly could do. And down here on the right, I have a lagoon. That will, excuse me, that will become important later when I talk about the work we've done with indigenous communities. Because they live in remote areas. We're talking about a community of maybe 500 people, small group of people in a very isolated area without active wastewater treatment. So the way they treat their waste is it just goes into a lagoon and they let nature basically decrease the biochemical oxygen demand and remove nutrients, but they don't have any active treatment. It integrates the whole population. It measures these asymptomatic people. It's rapid, effective, cost-effective. In a minute, I'll show you. We were collecting samples doing 24-hour integrated samplers. They're refrigerated, and they go for 24 hours, take an integrated sample proportional to the flow of the wastewater, and then we take that sample. It captures all of the individuals in the population. And the key thing that I'll show you later is it gives us a 7 to 10 day lead time on the waves of people that are going to end up sick. I'll show you the data later, but we could give the government, we could give the health authority, and I was advising the health authority every day. Once a week, I was on the television and for almost 2 years, telling people what was going on. But we would give them 7 to 10 days, and I could accurately predict how many people would end up in the hospital, how many people would end up in the ICU, and how many people would need a respirator. Because of that, then the government could move people around, they could move respirators around and hospital beds around to accommodate these peak flows. And it saved hundreds of lives. So I'm pretty proud of that. Now, there are some disadvantages. One is when the waste has gone into the wastewater treatment plant and gets mixed with all the other waste, it gets diluted. Okay, so now we've diluted our sample. So any of you that are chemists know the more you dilute the sample, the harder it is going to be to detect it. There are interferences. It's wastewater, right? There's a lot of shit in there. Oh, sorry. But it's wastewater, right? So it's complex and there's a lot of chemicals in there. So the wastewater treatment process in part is pathogens treat the waste. So it is basically doing that. It's removing them. And RNA is not very stable in the environment. It's not like DNA. And it gets sheared, it gets degraded. And so what I'm trying to do is look at that degraded RNA and try to determine whether it came from COVID or not and quantify it. When people ask me what we were doing and how we did it, I told them it's sort of like if I took a copy of the South China Morning Post and chopped it into 1 million pieces and spread it all over the campus, and then you asked me to tell you what the headline said, okay, from those little pieces all around the campus. So it's a difficult problem. And it took our whole team to be able to figure out how to do this. For the viruses, there are multiple variants mixed together. So it was important to know whether it was Delta or Omicron or these different variants that people were being exposed to be able to plot the waves of these things as they came. So we developed techniques to be able to quantify them and identify all of them. And then I'll talk about normalization. We need to normalize to a number, which is really the number of people discharging into the wastewater treatment system. We did a study on the day where the whole country in Canada does a census. So everybody fills out a questionnaire and they have people that go around and talk to people. Basically, we know on that day in every place in different regions how many people are there. And so we did a study with Statistics Canada and Health Canada on that day, and I got samples from 247 wastewater treatment plants to measure things that are in the water that we could normalize to, that would tell us the number of people discharging on a given day to the wastewater treatment plant. There are disadvantages. For some things, it's better and easier and more effective, and for other things, it's not quite as good. So there are challenges, and there's this matrix complexity that's an issue. But for the other things of overall cost and rapidity and integrated measure, it's much, much better than the old way of doing things. There, some of this was going on. In a minute, I'll show you our dashboard. So we had a dashboard up at the university that gave people real-time information on what was happening in the communities. That dashboard got hundreds of millions of hits. Hundreds of millions of hits. People that saw me on the television told me, oh, we're planning our whole week around what you tell us is happening with COVID. So people were using it. So one of the key things for me was not only to do the science and figure out a complex problem, but to help people and to do it in real time in a way that they thought was effective. An award from basically all of North America, the Good Cities Award, Effective Helping Cities. We won that international award. So anyway, 273 universities adopted this. So I was advising people at Notre Dame and all these other places that wanted to do this. 58 countries, I guess about at this point, about 3,500 sites, all over the world. And one of the good things that came of this was I was on video conferences continually during this process, and my internet at my house was not good enough. So I got, at government expense, fiber optic run to my house, which was pretty good. The government said, John, you can't keep getting dropped in these things. You're the chairman. So they put in fiber optic all the way to my house. So laboratory was the lead laboratory for the entire country. We were collaborating with the National Microbiology Lab, which is in Winnipeg, which is right there. And then these were all places across the country, including some of these remote areas I told you about, some of these indigenous communities up here in the territories. No roads up here. And I was monitoring in 3 cities around in this area, but advising all of these people. And one of the things we did for the program was I got samples from all these laboratories, and we did a validation test. So everybody that was monitoring and using the technology sent samples, and every week we would monitor those and tell them based on our standards whether they were doing a good job or not. Over the United States, And there were a number of things to figure out. So we've published probably 30 papers on this, on different aspects of it. Everything from sampling, how you do the sampling. So you can either sample liquid, water, or you can take sediment from the wastewater treatment plant. You can use adsorbents. It turns out cotton wool is a good sampler. So we came up with different ways to enclose it and protect it in the wastewater stream to be able to collect it. Compared those sort of solid phase samplers to our active 24-hour SIPR samplers. We looked at whether you should measure it in particulates or in liquid. So there are all these different issues of sampling and concentrating and identifying and normalizing all took research to do. So we did a lot of that. I'm not going to go through the whole thing. If you're interested, of course, I'm leaving this presentation and a list of publications that you can look at. So we worked up a whole pipeline of how to do this analysis, including using our chemical analyses. That I chaired writing for the government, where we published all of our techniques and all the quality assurance, quality control that you need to accurately do this. You need to make sure you're doing it correctly and getting accurate results because you're affecting decisions that affect people's lives. So this is public health and a lot of potential criticism if we don't do it right. So here's some of the reports. And on the other side is some of the data. So I would get samples from the different, I had 3 sort of archived ones, and then we would analyze those for all of the laboratories. So I would send them samples, they would analyze it and send them back to me. And then we've got laboratory A, B, C, D, E. When I wrote the report, I didn't say this is Professor Lung's lab. Okay, it was A. Kenny would have known what lab he was, but other people didn't know. Normalize, I told you. Figure out how many people were this N1 and N2 primarily for our quantification. So this is our viral load over here. These are correlations with over here the number of daily new cases in per 100,000 individuals. And so these are different ways to normalize. We did internal standards. You can then normalize to things like other viruses or chemicals. And so we measured a whole bunch of different chemicals, BOD, nitrogen, ammonia, all kinds of things that would be in wastewater that might be a good indicator of the number of people discharging. One of the things we tried was something called copper stanol. Copper stanol, Professor Lung shaking his head, he knows what it is. Copper stanol is an alcohol that's a degradation product of cholesterol. And so there's a lot of cholesterol in wastes, and the one we look at is specific to humans. Why is that important? We don't want to be measuring things that would be indicators of waste coming from cats and dogs washing off the street. So we just want people. Other ones that you can use are things like creatine, and that's pretty effective. But in a minute, I'll show you what we hit on, which some of my family knows, but they won't. The rest of you might not know. Indicators have to be quantifiable, have to measure it, and they have to have they have to be conservative. They can't be degraded, and they have to not be bound to particulates or the filter paper that you might be collecting samples with, or pipes in the system. And they need to be stable, and they need to be fairly constantly excreted. because we're going to try to use them as a surrogate to predict how many people we're discharging at any given time. Population census thing where we were calibrating these to all the cities across Canada to the population that we knew was there. Now, another interesting thing, I don't live in the city. I have my own septic system, so I discharge to that, so I wouldn't be counted. So that's an issue for rural areas. And we'll come back to that in a minute, how I handled that problem. Markers that have been applied. I wrote a review paper on methods for censusing in wastewater. So, you got social things. One that was suggested was cell phone use. We can get the data so we know where everybody is and how many people are there using their cell phones. I tried that. It does not work very well. And besides, there were a lot of legal social implications. People didn't want to give up data. They didn't want us to be gathering data that showed where everybody was at a given time. So we didn't do that for ethical reasons. But there are a lot of other things. And down here, we could use fecal monitors, things like bacteria, other phages. There are phages in bacteria. There's one called the mild mottled pepper virus, would probably be good here in Hong Kong or other areas on the mainland. It comes from chili peppers. And so people that are eating chili peppers will have that virus in their waste. And the nice thing about it is virologists know how to measure viruses, so they can measure it. If I go to a virologist and say you need to use an Orbitrap chemical analysis to get your monitoring normalization, they're not going to be able to do it. So you can use other viruses. We found that does not work as well as our chemical indicators. Correlation matrix of all these different kinds of things that you can measure and figured out which ones were the most predictive. And it turns out that it was this one right here, ACE. What is ACE? You know what it is? It's, Professor Leung, as usual, knows. It's a sweetener, artificial sweetener. And it's in soda pop. And it turns out a lot of Canadians drink soda pop. And in fact, they drink about 3.5 soda pops per day, per person. And we considered caffeine because a lot of people drink coffee or tea. And caffeine, I can measure. I get a nice peak about 9:30 in the morning in the wastewater. It comes through. People get up, have their morning coffee, and then about 9:30 they pee out the caffeine. And with caffeine, you have to actually measure a metabolite. You can't measure the parent material. And caffeine works okay, but it wasn't as good. And because this chemical is basically refractory, it does not degrade. Doesn't degrade in your body, and it doesn't degrade in the wastewater treatment plant, and it's water soluble, and it's released very constantly. It's down here on the bottom. So we could get really fine adjustment on an hourly basis of what was going through the wastewater treatment plant. And that's what we could normalize to. And when we did that, it really, really tightened up our quantification, the accuracy and precision, and made our models much, much more accurate. So if you have to do this kind of monitoring, my bet was it would be copper stanol would be the best one, and it turns out it's not. To do copper stanol, we had to use GCMS, and we had to do a derivatization, and it was difficult, and that added a lot of variability. So copper stanol, which had been used historically, was not the best choice. Oops, sorry. So this was the normalization, once we did that, we got very, very good R squared. So here's our p-value 0.001 in predicting the number of people, new cases per 100,000 based on our wastewater monitoring. Not too bad. Sites I was doing, we picked them because of their different size. These were the three that I was doing intensely and doing our studies on with all these different issues we had to work out. And they had different demographies. Some were more rural, some more urban. Saskatoon, my hometown, my house is out here somewhere, is the largest city in the province. We had a satellite lab where I trained the people how to do this that we're doing Regina, which was the capital of the province. So that was important. And then North Battleford is really an indigenous community, though big enough to have a wastewater treatment plant. Board. So this was up every week, updated every Monday morning. And then in the evening news every Monday, I was on the television. I also did radio interviews. I did them in English and in French for the whole country. And then people could call in and ask questions about things they wanted to know about. And so this was what was on the dashboard. I had it for Saskatoon, Prince Albert, North Battleford, and all of our satellite labs did the same thing. So remember, Mark Servos, my buddy in all of this, was in the Greater Toronto area and doing monitor in cities out there, and they also had dashboards. Data that we were getting. We were looking at different things. Remember, I told you that some was, Omicron or the Delta variant. So I had detailed data on a daily basis. Every 24 hours, we had an integrated sample for all of the waves of all the different variants. Of infections and hospitalizations and ICU occupancy. This is what our Office of Emergency Management wanted to know. They needed to know who was going to be sick. And when these are the waves. See, there's waves coming through here. So very low incidence of people getting sick. And then suddenly there'd be a big wave of people. And this is our data. So this is the lead time that I was telling you about. From the model that I developed, and it is very, very, very accurate. We can predict within 1 or 2 people how many people were going to be in the ICU. I could almost tell them about what time of day they would show up. So doctors were astounded by this. Because remember, they didn't believe it. When I told them, I'm going to measure things in wastewater and tell you who's going to be sick at some point in the future, and they just didn't believe it. They said, no, we have to get the sick person, we have to take a nose swab and do all these things. And so after a while, then they were really believers. They said, wow, that really works. And the result of that is now we do this for polio, we do it for measles, we do it for common other viruses like influenza, and it's being done routinely. And the doctors understand it, they know how to interpret it, and they know how to use it. So we've published our model. Your model would have to be calibrated to your city, okay? You couldn't just take our model. You can take the parameters in our model and parameterize it for your city, and it should work pretty well. And we published that with our colleagues. We had an index. So remember, we're dealing with doctors and nurses and the general public. So if I start talking to them about quantitative PCR measurements and that we had to look at this mixture and we had to make cDNA and then we had to amplify the cDNA, their eyes just roll back in their head because they're not scientists. So the director of our emergency services office that I was working with all the time said, John, we need something simple. We need something that people can look at and interpret in an easy way. So Marcus Brinkman and I put our heads together and we thought about this and thought about it and thought about it. And management people wanted, they really wanted yes, no. They wanted red and green. They said, tell us when we have a problem and tell us when we don't have a problem. Marcus and I couldn't really come up with a way to do that very well. So we came up with 4: low, medium, medium high, and high. And we based this on 2 things. We looked at a lot of things that we could put into the model, but I wanted it to be simple and easy to do and easy to interpret. And so it's color coded, so a doctor can look at this, and it's based on the rate of change and the relative concentration on a given day compared to the long-term average. So it's just 2 parameters. But the key one is the rate of change, R, when it goes in the model. So when you see red, it tells you you're much higher than the long-term average and it's changing fast. So you need to do something about it. If it's green, it's pretty low and it's not changing very fast. So you've got some time to react and wait. And if it's yellow, look out because it's coming. So you need to worry about that. How am I doing for time, everybody? Am I okay? Okay, good deal. Oh, get tired. Okay, now I want to come back around to the socioeconomic aspects of this. So we've got the cities handled pretty well, and I've taught the whole country how to do this, and it's up and running, and it's been turned over. A lot of the jurisdictions hired my former people. The province started their own monitoring program at cities we weren't doing, and they pirated away one of my postdocs, Femi Ogulala, to run that whole program for them. So things were up and running pretty well. We had it all standardized, and we knew how to interpret it. So now I could turn my attention to the indigenous communities, to our Aboriginal populations. Now, interestingly enough, they look like you guys. Why? Because 10,000 years ago, they came across a bridge from Asia. And so these are people that have been in North America for about 10,000 years. And they have their own cultures, and they live in very remote areas. When COVID came, one of their responses is when someone in the community gets sick, they do what's called a smudge or a pipe ceremony. So if I got sick, you would all gather around me and we would take a tobacco pipe and everyone would pass it around. What are you thinking about COVID? Henry's smiling. What's the worst thing we could do if Henry's sick? Is all go over and gather around Henry and smoke a pipe with him, right? It's probably the worst thing you could do. But culturally, that's what they do. And they have a community center, a lodge in their community. And when people were sick, they would all get together to support that person. So culturally, it's very nice. From a human health, public health perspective, it's a disaster. And what happened was these diseases, when they would go into that community, would just rip through the community and everybody would get sick and people were dying. Now, they're remote areas. That means they might have a nurse, but probably not a doctor. They might have a small clinic, but not a hospital. In these communities, when someone gets really, really sick, we bring them to Saskatoon in a helicopter. Okay, we go out and we get them and bring them. When a whole bunch of people were getting sick, that just wasn't possible. Now, what else did I tell you about these communities and the government? They don't trust the government. And so when we started working with them and trying to figure out how we could help them, the first thing they said was, John, we trust you. don't ever share our data with Canada Health. We do not want the government involved. We don't like the government. We don't trust the government. So don't even talk to the government, because we're not going to talk to the government. So what do? What I did was develop this system. We call it a gene expert system. What it does is it's a cartridge system that will allow you to do the measurements without needing a high-speed centrifuge or qPCR instrument like we have in our laboratory. You don't need to sequence things. It's very, very, very simple. And I convinced the federal government to buy 35 of these for me. And I took it around to all these communities and told them what it did and showed them how it worked, and then mostly trained high school kids, high school students to run this. And they were really excited, and they did a really good job. And at the same time, what was a side benefit, I guess, of this was these kids were developing an understanding and a love for science, which they never had before. And that they wanted to get more education and leave their community and go to university, which they never did before. So it was a really, really nice benefit. Can do influenza, flus, a whole wide range of flus every year, different one. RSV, I don't know if anyone's had RSV. I had RSV, it's terrible. I coughed for weeks and weeks and weeks. Polio is resurgent. You may know in North America, there are anti-vaxxers. These are people that don't believe in vaccination. And that means that they don't get their kids vaccinated, and polio is coming back. And we just this year in Canada had an epidemic of measles, which had been basically wiped out. But now, because people are lazy or they're anti-vaxxers, they don't like to get vaccinated or have their kids vaccinated, some of these diseases that we thought we had eliminated are coming back. There was a scare with monkeypox. Anybody, I don't know if you read about it here, was a new virus that was showing up in Canada, and the concern was it would be like COVID-19 all over again. It turned out it wasn't that big of a problem. It's not as easily transmitted, but we were set up to monitor for monkeypox as well. You get your sample, you just put it in a tube, you put it into this cassette, you push the button to start, and it runs through a cycle, and it gives you the data. So it's very easy to interpret. It could be used for lots of other things as well. Now, it's pretty accurate. I wrote a paper where we've calibrated it to our more detailed, more sophisticated qPCR methods, and it's pretty good. It's pretty accurate. It's not quite as sensitive, but it works. When we do for any of you that are familiar with qPCR, you have cycles, right? So we're copying, we make cDNA and then we copy that, and every cycle you double. So 2, 4, 8, 16, 32, 7, it's going up. My detection limit for COVID that I can report is a CT value of 35. What that means is 35. If I have to do more than 35 cycles, things get so variable that I can't really report it, because you have a little bit of variation at the bottom, and by the time you amplify it up, the error terms just get huge. So anyway, our limit's about 35. And these instruments can just about approximate that without any pre-concentration. In the lab, I pre-concentrate things using high-speed centrifugation and a protocol we've developed to isolate the RNA. And this is sort of some pictures of it running. We put these out in these communities. Remember, now they don't have wastewater treatment plants, but they have lagoons where their waste is is going. Things that happened in this process. As I was meeting with these communities, and remember they trusted me, but they didn't trust the government. So we were training them, and I was in the community. And I remember in one community, one of the elders wanted to talk to me alone. And so we went off, and he said, "John, we have some concerns." I said, "Yeah, okay. What's your concern?" And he said, "We want to protect our people. We want to protect our kids, but we don't want to kill the virus. It has a spirit. And is there a way you could keep the virus alive?" And I said, "No." Anyway, interesting social interactions. But there is some maintenance involved, but it's pretty easy. We were able to train high school kids to do it, and they were very, very conscientious. I was really impressed. So this is really all you have to do. Hopefully we have some time for questions. Kenny knows what I know and what I don't know. So if you have other questions about other things that I might know about, I'm here, for the week, Henry? The rest of the week. My room is 5422, and if my door is open, just come in. With that, I'll stop.
So wonderful. Thank you, John. Now I would like to open the floor both online and also in the room to have a dialogue with John. Any questions or comment from the audience and also from those in Zoom platform? Yes, John, please.
Thank you, John. It's really I have no question now. Okay, thank you for the excellent talk. It make me recall it's a very hot topic actually at the pandemic period that to identify the virus in the wastewater. So my question is, because I see you also show the quantification process of the virus in the wastewater, but do you think because different wastewater treatment, the technologies used are different, do you think the results are comparable between different plants, or you just see the changing trend in the same wastewater treatment plants? Thank you.
Good question. So he wants to know if this can be generalized, I guess, among different wastewater treatment plants. And it's true, there are very different types of wastewater treatment. We did studies all across Canada, and each one a little bit different. And you really need to calibrate it to your city. Okay, it worked pretty well. The model that I showed you that we developed was based on data for the whole country. So it works pretty well. What happens is the absolute amounts vary among locations. And for instance, in Montreal, they have a very different collection system. And we did a lot of work about where in the system to collect. Some of the work we did was, should you take raw effluent coming in or influent coming in? Should you take effluent going out? It was probably better to do the inflow as opposed to the outflow. There are different places within the treatment. We found that if you take your samples in the initial mixing tank, it's the best place before you've hit a lot of the steps that might be different in the wastewater treatment plant. We looked at whether you should use the particulates. The advantage of particulates is there's more RNA in the sample. It's concentrated, but at the same time, it's more degraded. So if we took it from the settling tank, we got higher concentration total, but more degraded. So it's a trade-off between the two. And in the end, we opted to take particulates from the initial settling basin, worked about the best. that minimize the effects of the downstream differences in the plant? Yeah, very good question.
May I supplement what have been done in Hong Kong?
Sure.
The method similar to yours developed by Tong Jiang from HKU. And then in Hong Kong, we are really densely populated city. Instead of doing the sampling from wastewater treatment plant, they're doing district-wide sewage collection system. Then they open the manhole and then get sample. It's really effective. They know where the virus come from, like substrate pole, they can point out because not Team Sartre. Then they actually really close the whole substrate pole, and then everyone need to do the test on spot, and then not allow them to move out during the time. But the result really good. Give the hospital authority 3 days in advance about the outbreak. Then because EPD paid Jiang Tong to do this research, and then EPD got the IWA award of the years.
Yeah.
Yeah.
And along those lines now, something else. So we can do the same thing. In fact, in many cities, we did that. We sampled at different, we call lift station, but different manhole you can put sampler in. And there we were using what we call torpedoes. They're little plastic containers, and we put feminine hygiene tampon in. And they worked really, really well. They're uniform size. They were sterile to start with, and a lot of advantages. Sounds silly, but it worked. And so we were doing that. But we had a big discussion. Remember, I was the chairman of this whole committee for the whole country, and a lot of discussion about the ethics of sampling. In my city, the west side of the city is predominantly indigenous people, okay, native peoples. They live in the city, they've moved to the city, but they tend to live together on the west side. It was decided that we would not separate the city into sections for ethical reasons because of the stigma of saying, oh, the west side is more contaminated, or the north, so we chose to not do that. But on the campus, we did specific buildings. So we used the technique, and it is very effective. But in our situation, it's an ethical decision of how you do that. If your population's fairly uniform and you don't have to worry about stigmatization of particular ethnic group or socioeconomic status, something like that, it works very well. But we were concerned about that. And in part, it's because I told you that the distrust of the federal government of that group of people. And basically, they didn't want anybody to know if they were sick. So it was a difficult socioeconomic decision. Yeah, good point.
Stigmatized. So very, 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 very different society. Here is more, how to say, get things done rather than campaign.
Yeah, absolutely. Yeah, and you have to work with remember, I worked with all these different communities, and every one of them was different in what they wanted, what they would accept, how you had to teach with them. I mean, everything, it becomes important to tailor it. The work we did in Montreal, I speak French, and they speak French in Montreal, but they speak something called Québécois, which I speak Parisian French, and we can hardly communicate. But they had a whole different way of thinking about things, about what they wanted, how they wanted to do it, and what they would accept. And we had to adapt it to what was right for them. Yeah, excellent point, Kenny.
See if we have any questions from online. Yeah. Chris, do you have any questions or just say hi to John? Chris Wong from Betty Sue, I guess. I would, yes, please, Harry.
Say you have a DNA team, so you start with the RNA for COVID. But have you considered measuring the spike protein in the wastewater? So what the merit, pro and cons, comparing measuring spike protein and COVID RNA?
Well, we did measure spike protein. We did most of our quantification based on the N1 and N2 structural proteins. The reason for doing that is they were more conserved, so they gave us a good number for total COVID. What I didn't get into was those charts had different variants, and that was done from the spike, which was changing very fast. So I wanted to do both. I wanted to have a constant measure that I could compare among the strains without that changing, but I also wanted to identify whether it was from this strain, and we used the spike protein for that. So I was always measuring spike protein, but I was quantifying for the total COVID there on the N1 and N2 structural proteins because they were more conserved. And they're also more stable in the wastewater. You remember, RNA is not very stable. And so that's a good thing because then in the wastewater treatment plant, you pretty much destroy it. But to sample it and be able to identify it, because there's all kinds of other RNA there, RNA from lots of things, not just COVID. So I had to separate and identify and then use a set of primers that would amplify only what I wanted to look at without amplifying everything else. Because if I made cDNA on all the RNA and amplified all of it, I have such a mess I couldn't figure out what it was. So it wasn't an easy problem to solve. Other people around the world maybe didn't do it in quite as sophisticated a way to be able to determine the differences in the variants. So one of the papers we've written is the timing of the succession and development of the variants. And I was also doing sequencing through this whole time. This was just for quantification, but we were also sequencing and using that data as well. Because we were interested in if there were going to be new variants. One of the big concerns of the committee I was on is these variants were coming at us every couple months. And it was like, is the next variant going to be more virulent? And if so, what is it? And we need to be prepared to be able to monitor for it. So we were doing sequencing all the time. And I was sharing samples with our national microbiology lab, and they were just sequencing the piss out of these things. Because they have much more sophisticated automated sequencing. Yeah, no, but we were looking at the spike protein for sure.
From your talk, you use a sweetener as the denominator to calibrate for the seasonal variation. From a Chong study to look at pharmaceuticals in 190 estuaries around the world, he done 80% of the sample. Now we find sweetener is the top pollutants in all estuaries, number one, right? And very interesting.
Why? Because
diabetes.
No, because it's persistent. It doesn't degrade.
Yes, yes.
And that's why we liked it. And it wasn't just for seasonal differences. We were doing it on hourly differences.
Oh, wow. So I'm thinking we should make a rapid detection because sweetener can tell you whether this is polluted river or not.
Exactly.
Yeah, that should be the next invention we should develop.
Yeah.
Last chance. Any question beyond the talk? Yes, Professor Tong.
Thank you, Joe, for your very interesting study. So actually, I have no question. So it's like I also can share some experience from Singapore. So because previously, the team actually in Singapore, I work from the Singapore, also lead the similar studies in Singapore. So and also they do the continuously monitoring of for the COVID, and they also collect the sample from manhole. And also they installed the
From where? Tom?
manhole.
Manhole, yeah.
Yeah, and also they installed the auto sampler. So and continuously to collect the samples. And after the COVID, so because COVID time is over, and they talk to the National Environmental Agency in Singapore. So I think now they are monitor the antibiotic resistance for the AMR. So they continuously to detect the AMR work. And also I have a friend in the Japan, so in Tokyo, the University of Tokyo. So actually they establish a center and to do this kind of wastewater surveillance, and they collect a lot of samples from the airport. So actually they have a lot of data and also to share very good news because they can use this kind of things to do the early warning of the different virus. So I do believe this is a very interesting project and also can do the continuously support for this kind of work. Okay.
You know why they do it in Singapore? Because my former student, Shane Snyder, was the head water guy in Singapore. So he talked to me, and so, yeah. And they did a lot of really good work. And we've compared our information with Singapore as well. Yeah, it can be very, very effective. Not easy to do.
Because I see in Singapore, previously before the COVID, actually they already have one kind of this kind of project.
Right.
Their main focus is antibiotics resistance for the AMR. And because the COVID come and they can convert these kind of facilities and to detect the COVID. And after COVID, we know AMR still a very important issue. So that's why they go back to the AMR work. Yeah.
Right. And we're also doing that right up until the time I retired. And my last postdoc was working on antibiotic resistance and looking at it relative to various not antibiotics so much, but what's the word? Chemicals. I can't think of the right word in English. Not antibiotic drug, but disinfectant. Yeah, disinfectants, because that we found was highly correlated with development of antibiotic resistance. And so we've been doing that. In fact, I was just communicating with Charles, my postdoc, by email this morning about a paper we're writing on just that topic. So and antibiotic resistance is a huge concern. I have personal experience. I had a sebaceous cyst on my back, very simple thing, but it got sort of big and then it broke open. They did some surgery on me, and in the process, I got a drug-resistant bacteria in my spine. Put me in the hospital in Europe, in the Netherlands, for 2 months. It almost killed me. And I was on antibiotics, a cocktail of antibiotics for 2 months, and it killed my kidneys. So it's personal with me. This antibiotic resistance is a real issue. And I'm too old to figure it out, but some of you young people, I would encourage you to work on it and try to solve it, because it's going to be a real big problem, for sure.
So, yes, please, last question.
Thanks, Professor John. And I have a small question, because the people's experiment for everyone is the fixed quantity. So if when you calculate the population or the for RNA, because in the morning I can pick 10 kilogram and I can to go to another state, other point and to just other building to point 10 milligram. How to calculate the list quantity between the belongs to the one people, but maybe there are 3 people fixed out. you calculate it, that's this belongs to 3 people. But when you calculate this, the data you calculate belongs to 1 people. There's the difference between the normalization. When you predict a normalize, maybe the population is very high. It's different.
Right. So it's large numbers. You notice I was calibrating to number of people per 100,000.
Yeah.
It wasn't individuals.
But during the COVID-19 period, people always they cannot to go anywhere. They can to stay in home in the very small area. So we can to calculate this with the water sample is very stable for the population. But but now everyone can go anywhere all over the world. They can travel and they can do everything. If you how to make sure that this data belongs to a stable data of the population?
I'm not sure I understand your question, but everything was for big numbers. It was just a correlation.
Yeah.
We're not measuring individual people.
Yeah.
And you would not really be able to reduce this kind of monitoring down to individuals.
Yeah.
What we did do was reduce it down to long-term care. One of the problems we had were with elderly people in home care. If COVID would come there, they would die. And so we wanted to make sure they weren't getting infected. We didn't want to wait until it was to the point where they were going to die. We wanted to know early on if there was somebody there that was infected, and then we could close down that whole facility and monitor everybody in there on an individual basis. But none of the other monitoring was ever meant. We looked at airport monitoring, and in the end, we decided it probably wasn't going to be very good. One of the concerns the government had was people flying around the world that were infected. And could we somehow use this at airports? And in the end, I determined we could not, because the lead time we'd have to give to be effective relative to someone flying around the world wasn't long enough. We'd have to have things done on a minute basis to be able to say, oh, that plane. What the government wanted me to do was be able to monitor like a plane and then decide should the plane go or not go. In the end, I told them I can't do it.
John, I think Xiaotong's question is more relevant to how to do the wastewater surveillance in a more dynamic communities. For example, in Hong Kong, someone may do smoke or weeds in some area and they go back home to sleep. So how can you calibrate these movements? So
you can't.
You cannot, right? Yeah.
No, you can't. And so, okay, thanks, Henry, for sort of clarifying that. We wanted to look at that because we were concerned about people on vacation moving around or deciding that, oh, okay, I'm supposed to not leave my house, but they were leaving their house. One of the things we wanted to do was use cell phone data for that purpose. That gives you individuals. For what we were doing, it was decided that it was unethical to do that. Okay, that's why I showed you the data. We looked at cell phone.
Also use our octopus cut.
So remember, I was the chair of a national committee dealing with Health Canada, Statistics Canada, the National Microbiology Lab, and we had lots and lots of layers of government officials involved. And when I said, hey, we could use cell phones, they said, no, you cannot do that. Because in Canada, we have very strong privacy laws. And they said, you would violate the privacy law, the standard law of Canada. You cannot do that. So we didn't do it. But you can. And that, but still, the idea is here, you're integrating a big population. It's never going, if you want to do that kind of monitoring, then you have to go back to something else. This is not going to work for that.
Wonderful discussion. So we have to close because we have to head over the classroom to another course. Can I invite everyone come forward and take a good photo with John together? Come to the front, please.